The overall goal of the following experiment is to measure UV sensitivity by determining the minimal erythematous dose in fair-skinned mice pre-treated with forskolin. This is achieved by first preparing the dorsal skin of mice to be irradiated by shearing and epilation. Next, topical treatments of forskolin are applied to the dorsal skin. Then the mice are exposed to different doses of UV irradiation to determine the effects of forskolin-mediated melanization on UV-induced inflammation. Results are obtained that show that topically applied forskolin robustly increases skin pigmentation and that animals with pharmacologically enhanced melanization are UV-resistant as determined by MED.
These are proof-of-concept studies that show that it is possible to pharmacologically induce a tan in an individual incapable of tanning because of a defect in a very important melanocyte gene called the melanocortin-1 receptor, or MC1R. In human populations, their loss of function of MC1R correlates with UV sensitivity, sunburning tendencies, and inability to tan, and later in life, up to a fourfold risk of melanoma. We chose forskolin as a drug for these studies because it's able to bypass a defective melanocortin receptor signaling pathway and trick the melanocytes into thinking they have a good MC1R signaling pathway, and therefore they make a lot of melanin, which gets deposited in the skin and protects the animals against UV radiation.
Though we know this method provides insight into skin darkening and sunburn, it might also be important for cancer prevention, since we know that UV radiation is the major carcinogen for the great majority of skin cancers.